right, hi everybody. Today we are finally going to be talking about regulation of gene expression. This unit 6, is called Gene Expression and Regulation. Remember that regulation is a fancy way of saying control. It's a way that we turn our DNA on and off. Remember the whole analogy of the babies having hairy armpits. We don't get hairy armpits until later in life because our DNA has been activated or inactivated depending on what's happening in our bodies. Same thing, every one of my cells in my entire body has all the DNA for every body part, but I'm not growing a liver right here. I'm growing skin. Why am I not growing a liver here if the DNA for liver is right there? Because the liver genes have been shut off. So today we're going to talk about how prokaryotes turn their DNA on and off. I try to get that phrase, regulate their gene expression. How they regulate their gene expression. We're going to start with prokaryotes because it's so much easier, and this is hard, so much easier than the way eukaryotes do it. We'll save eukaryotes for next time. All right, so give me just a second to get started here. Oops. Okay, in prokaryotes, just a quick reminder that prokaryotes, because they don't have a nucleus, because they're tiny, they can couple transcription and translation at the same time. So it makes their nucleus it makes their process of gene expression super efficient and requiring a lot less different controlling factors that we're going to see in the eukaryotic notes. So this is an image we've already seen. This is the prokaryotic DNA. So remember that they have circular chromosomes. So you're not seeing the circle here. Each of these guitar picks, which you're going to see again in just a minute, those are RNA polymerases. They are reading one side of the DNA and making the mRNA, and that's the red ribbon. And then because this is a prokaryote, the mRNA doesn't have to leave a nucleus. Right away, the ribosomes grab on, and a whole bunch of ribosomes can do it. We call that polyribosomes. And they're immediately building the proteins that this mRNA is coding for. So that's the process happening super fast. So we're going to talk today about positive and negative control of the of two systems that are super famous in a prokaryotic cell. So the things that we're going to talk about are called operons. Operons are clusters of genes that contribute to a particular metabolic task. So today, the first operon that we're going to talk about has to do with how to digest lactose. The second operon we're going to talk about has to do with making an amino acid called tryptophan. This is found, operons are found only in prokaryotes. Eukaryotes are a lot more complicated. And so there's two types of operons. There are inducible and repressible. And you're going to see that the lac operon is inducible, which means you induce it or you start it or you trigger it. And repressible means you stop it. It's always going, but then sometimes you can stop it. And that isn't what positive or negative feedback has to do with. All right, so let's take a look at the first one, the lac operon. So the phrase lac means lactose. I got a best way to start this. Prokaryotes want to take lactose, which is a disaccharide, and they want to break it into its component sugars, glucose and galactose. They need glucose and galactose for cell respiration. So when they're hungry and there's not a lot of glucose available, they want to be able to digest lactose. So this is a series of genes that controls whether or not they're going to be digesting lactose. Keep in mind that they don't want to be making the enzymes for digesting lactose if no lactose is available. That wastes the prokaryotes' energy, and they have this much energy, they can't afford to waste their energy. So they're only going to make the enzymes if lactose is present. So let's take a look at how this, and you need to study every aspect of this diagram, and you need to be able to understand every little corner of this diagram. So this is an example where lactose is absent. So let's say we have a group of E. coli bacteria, and we drop them into a bowl of water. No lactose available. They don't want to be making the enzymes for digesting lactose because that would waste energy. So let's take a look at the operon itself. So they're showing you here that this represents DNA. You cannot see the A's, T's, C's, and G's, but this is the prokaryote's DNA. So that's what it's representing. So there's a gene upstream, they call it upstream, that produces a repressor. That's called a regulatory gene. So the regulatory gene makes the repressor. The repressor, they often draw as a funky shape, like a puzzle piece or maybe a Lego block. And the repressor fits right onto the DNA itself. It clamps on like a roadblock. So I use those R's to try to help people remember. The repressor is the roadblock. So the promoter is the area on the DNA that promotes transcription. It tells the RNA polymerase, this is the beginning of the gene. You should come here and start transcribing. So the RNA polymerase pulls up. Imagine it's like a little car for our analogy for a moment. And instead, it can't get through because there's a roadblock. So beep, beep, it wants to get through, but it can't. So right now, the gene is shut off. We have repressed it in this case. So it says here the repressor protein made by the LAC-I gene is able to attach to the operator right here. It blocks it. RNA polymerase cannot transcribe the structural genes. These are the structural genes, and there's 3 of them following. It can't do them. Since the cell needs to be able to digest lactose, or that the cell needs to be able to digest lactose, since there's no lactose, no need to transcribe them. So this is how things remain in the cell as long as there is no lactose present. But what if we scoop those E. coli up and instead dump them into a bowl of milk? So now they are interested in breaking that lactose into its component parts. So they do want the enzymes to be formed. So in this case, the lactose itself, it's actually called allolactose in this circumstance, acts like an inducer. So you need to know the word inducer. An inducer touches the repressor. Remember that the repressor, I'm going to scooch back over here. Remember that the repressor has been sitting on the operator and it's the roadblock. So now when I put these E. coli into a bowl of milk, one of the allolactose molecules, it's related to lactose, is going to touch that repressor, and they cause the repressor to change shape. And when the shape changes, it falls off of the operator, and now our RNA polymerase, the little car, is no longer roadblocked, and it can start moving across. It's going to read the LAC-Z gene and make the mRNA, the LAC-Y gene, make the mRNA, the LAC-A gene, and make the mRNA, which are then going to be translated by the polyribosomes into the 3 enzymes that digest lactose. So it says here lactose is present, the repressor is inactive, the operon is on. We have turned this DNA on. So just to review down here, get my picture out of the way. The inducer molecule called allolactose, a form of lactose, binds to the repressor protein. This changes the shape of the repressor protein so that it cannot attach to the promoter. RNA polymerase is then able to transcribe the structural genes that the cell needs to be able to digest lactose. And this is how things remain in the cell until the lactose is digested. So the question is, what kind of feedback is this? In this example, this is called negative feedback because they are both switched off by a repressor. So this is an example of negative feedback. It's going to get tricky because in a few minutes, I'm going to teach you about a positive feedback that still has to do with the lac operon, just to make it difficult. All right, so let's learn. That was inducible. We induced it. We started the process when the repressor was pulled off. That's an inducible operon. This is a repressible operon. We can repress it. We can stop it. Normally, it's on, we can shut it off. The lac operon, normally it's off and we can turn it on when we need to. Or the E. coli, not me. I can't turn it on. All right, so it says the trip operon is what synthesizes tryptophan. So if you remember from our last set of notes, we talked about Beadle and Tatum's experiment where they were mutating bread molds and they were looking at the arginine pathway. It was a whole bunch of enzymes that worked together to take a precursor molecule and to form an amino acid called arginine so that it could build some of its proteins. Well, this is the same thing. Tryptophan, the bacteria has the ability to make tryptophan, and it requires a whole series of enzymes to take a precursor molecule and form into tryptophan. So when it's low in tryptophan, it needs to be making tryptophan, otherwise it can't make some of its proteins. It's one of the 20 amino acids. Tryptophan is one of the 20 amino acids. However, if you happen to place E. coli in a solution that is high in tryptophan, there's no need for it to make it. So it's going to want to turn this whole process off. So normally this series of genes, the operon, is on, but we can turn it off if it's not necessary. So let's take a look at the diagram again. It's DNA. This is all DNA that we're looking at. Here's the regulatory gene that makes the repressor, but normally the repressor is off. It's only going to clip on if we want to shut this process down. So notice that the tryptophan, the trip operon is on. It's activated right now. Here's our RNA polymerase CAR. It has not been roadblocked, so it's going to move across. It's reading the DNA. It's making the mRNA, which are leading to the production of these enzymes that make tryptophan. So down here, it just says when tryptophan is absent, the operon is on. The repressor protein is unable to attach to the operator. RNA polymerase, our CAR, can transcribe the genes that the cell needs to be able to synthesize tryptophan. So the whole thing is on. This is how things remain in the cell as long as there's no tryptophan present. However, if tryptophan is present, it acts like a co-repressor, which means it touches our protein and puts it into the active shape. It now acts like a roadblock. It gets in the way, so it's sitting on what's called the operator. The RNA polymerase, our CAR, can no longer get through. Beep, beep, let me through. Nope, won't let you go through. Why do we have it turned off right now? Because tryptophan is in the environment. It doesn't need to be synthesizing tryptophan. That would be a waste of energy. So it says when tryptophan is present, the operon is shut off. The co-repressor molecule tryptophan binds to the repressor protein. This changes the shape of the repressor so that it can attach to the promoter. RNA polymerase, so the promoter is a part the operator is a part of the promoter. So this little yellow area is called the operator, but the whole section is called the promoter. You can see that better in the lac operon diagram than right here. RNA polymerase cannot transcribe the structural genes that the cell uses to synthesize tryptophan, and that's good because we don't want to waste the energy. And this is how things remain in the cell until tryptophan is no longer present. So, whoops, I went the wrong way. So this, again, is an example of negative feedback because we are stopping the process. We are not allowing the process to excel. We're stopping the process with a repressor. So now is where I'm going to get it confusing. Now we're going to talk about positive control of the lac operon. So both the lac operon and the trip operon are both negative feedback or negative control. Now we're going to talk about positive control. So here's the issue with the lac operon. Let me get my picture out of the way. If the E. coli, okay, why does the E. coli want to digest lactose? It's to break it down to get the glucose and the galactose so that it can do cell respiration. But what if the lac operon already has plenty of glucose and doesn't need the lactose to be broken up because it's already swimming in a sea of glucose? Well, then we want to control things a little bit differently. So we're going to start imagining that the cell is hungry. We're going to say there's no glucose present, and we definitely do need to break that lactose apart. This new element in the lac operon is called the cap protein. So let's take a look at it. So it says lactose is present, but glucose is scarce. So in other words, this E. coli is hungry. It wants to turbocharge the breakdown of lactose, wants to break down that lactose as fast as humanly possible. So it says low glucose means the amount of cyclic AMP is high. Just trust me on that. The cyclic AMP or CAMP binds to CAP. This is the enzyme that's going to turbocharge our CAR, our RNA polymerase. So this is CAP right here. When they are together, when both of them are touching, the CAP attaches to the RNA polymerase. So it says the activated CAMP-CAP complex, there it is, right there, increases the rate of transcription of the lac operon, boosting the rate of transcription several fold. So now, instead of our RNA polymerase, which we learned about in the lac operon a few minutes ago, instead of it driving, I don't know, 25 miles an hour down the road, now it's driving 75 miles an hour down the road. And it's transcribing the 3 genes for digesting lactose super, super fast. So I like to think of CAP, or the activated CAP, as a turbocharge. It's causing us to step on the gas. This is an example of positive regulation. We are positive feedback. We are increasing the amount of transcription that is occurring when CAP is present. So now, now let's imagine that the cell already has lots of glucose. What do we do now? So in this case, there's not a lot of cyclic AMP, so CAP remains inactive. So it doesn't turbocharge the RNA polymerase. Now, the one thing I don't like about this diagram, the RNA polymerase should still be there, and it should still be driving 25 miles an hour down the road, but it's doing it really slowly and casually, and we're not turbocharging it. We're not doing it super fast. Why aren't we? Because glucose is present. We don't need to digest, or the E. coli doesn't need to digest those lactose super fast and get the glucose from them. It's already got plenty of glucose. Normal glucose level means the amount of CAMP is low. CAMP is not bound to CAP. CAP is inactive. Very little lac structural gene transcription occurs. So we're just going to drive 25 miles an hour instead of full speed. And that, again, is an example of positive feedback. All right, ladies and gentlemen, that's prokaryotes. That was the easy one. We're going to talk about eukaryotes next.